This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 973, recorded on January 6, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And it is sloppy weather here. It's snowing, but it's not sticking, so it's just wet and cold and yucky.
We have 9 degrees and cloudy, no snow here in New York City. Also joining us from Madison, New Jersey, Brienne Barker.
Hi, great to be here. It is 47 Fahrenheit here, cloudy, and I honestly think it has been consistently the same fog for the entire week.
Yeah, it's been foggy. Brienne is about 13 miles from me. We have the same weather, more or less, where I live anyway, not where I am now. Our guest today is from Tucson, Arizona, the University of Arizona, Felicia Goodrum. Welcome to TWiV.
Thank you. It's great to be here. I'm really excited to be here. It's about 65 degrees here and beautiful, as it always is in Tucson, so you should all be here.
Well, you don't want everyone there because it's not good for you, right?
It would be fun.
Couple months I'll be there.
That's right.
What are you doing? You going to do a seminar thingy or what?
No, my family spends some time there, and I go visit them over break.
Oh, okay. Very good. Get some
that's
sun.
Fantastic.
Yeah, I remember that from last year. Well, actually, Felicia, you and I talked many years ago about coming on, and it never happened for whatever reason. And then I saw you last summer at ASV, and I said we should have you. And so that was June or July, and this is how long it takes to do these things. So you're the first of our 2023 guests. How about that?
Yeah.
That's great. 2023 has kind of started off a little rocky, so this is a great way to change course, do something fun.
Yes. Before we start, I want to tell everyone that there is a research assistant position available in Amy Rosenfeld's lab at the FDA. This is the Center for Biologics Evaluation and Review at the FDA. Her laboratory is interested in understanding how cross-species anti-enterovirus antibodies can affect pathogenesis of infection. This would be for a research assistant to work on picornaviruses like Entero68 poliovirus and human rhinoviruses with genetic, biochemical, and cell-based studies for understanding multiple aspects of immune dysfunction associated with infection. The individual will do experiments in cell culture, participate in the development of small animal models for studying virus infection. Will work BSL-2. We will put a link in the show notes to a PDF where you can see more information and get Amy's email and email her in case you are interested. All right, back to you, Felicia. Let's start with a bit of your history, starting where you're from.
I'm actually from sort of a rural southwestern portion of Virginia, Roanoke, Virginia, and grew up there and became interested in science, and then ventured out from there. I went to Virginia Tech for my undergraduate, and then down to Wake Forest University, where I did my PhD in adenovirus with David Ornelas at the medical school, and then joined Tom Schenk's lab for my postdoctoral work in Princeton, New Jersey, before coming out to Arizona, which was a bit of a wild card. Never expected it, never planned it, never actually intended to stay, but just fell in love with Arizona, the people, the culture, the environment at the university is fantastic. So
I've been
here ever since.
How did you get from adenovirus as a postdoc to what you work on now?
Well, of course, so Tom Schenk studied adenovirus for a large part of his career, and he had just switched to cytomegalovirus. And everyone I knew, it's just such a thrill going to the DNA tumor virus meetings and interacting with all these people who had trained with Tom. They were such a cohesive, amazing bunch of scientists, and it was so much fun that I thought, I want a little bit more of that. And he had switched to cytomegalovirus because he thought he'd probably given all the interesting projects away to his postdocs. He's super generous in his training like that. And so because he had switched, I decided to join his lab.
Cool.
And, in fact, my graduate advisor was the one who said, when I was thinking about postdocs, he said, you should think about cytomegalovirus. I'm like, okay. I go off to the library. I come back. I'm like, they don't know anything about this virus.
And everybody has
like,
it.
Exactly.
It's not some obscure thing.
Has
it's listeners, you probably are walking around with CMV.
Exactly. But, for all of us that study viruses, a lot of what the allure to them is the pathogenesis and the disease they cause. CMV doesn't have that unless you're immunocompromised in some way. And so a lot of people are really surprised. Why do you study this virus that doesn't cause the disease? And, well, there is a disease when you're immunocompromised. But I think what we learn from this virus, the cell biology that it teaches us, is like so exciting.
Well, and
that's what has kept drawing me.
The question I would ask there is, why doesn't it cause disease?
Exactly. I mean, isn't that super exciting and interesting? And it's really, this virus has co-evolved with us for all of our evolution. And I think it just knows our biology so much better than we do. And this is the way that this virus has evolved to be able to persist is it's a trade-off and doesn't cause disease. So.
I would say, Brianne, that why would a virus cause disease?
Yeah.
No, exactly.
Why aren't they all like this? Yeah.
Yes.
Exactly. Now, this is a really crafty measure. But, it makes it such a complicated virus that has a genome that's 236 kilobases, and it encodes well over 200 proteins. There are definitely 200 open reading frames, but many of these open reading frames encode for multiple proteins. And so it's been estimated, somewhere between 200 proteins and 750, which is just, remarkable for a virus.
So you mentioned that everyone has this, everyone is likely infected with it. When are people infected and how?
Usually as children, and once a child is infected, they will shed the virus in their saliva and their urine for months to years before they get a really, truly quiescent latent infection in which they'll then carry the virus for the entire rest of their lives. And so I think, that's a really interesting piece. And it's one of the things that does actually result in a lot of CMV pathogenesis is that often women will contract an infection from their toddler, because, the interaction with those bodily fluids is pretty intense all the time. And then if that woman is pregnant, the second child can get a congenital CMV infection. And that's where this virus can be really devastating. And about 1 in 150 babies born in the U.S. have a congenital CMV infection. 20% of those will have some really severe developmental issues, cognitive issues, hearing loss, and the most affected children will die in their teens. And so it's really tragic. And so there's a huge effort to understand the congenital infection. And this is a place where we're learning a lot of interesting immunology, because you can be infected with CMV many times in your life, and your immunity is not necessarily protective to multiple subsequent infections.
Is there anything you can do if you're pregnant and you find that you're CMV positive?
Well, it really varies state to state. A lot of women are tested at the time of pregnancy for their CMV serostatus. If they're CMV seronegative, they may be followed to see if they seroconvert. There's not a lot. There is some use of ganciclovir and antivirals like that. But, the most important thing, where I think it's so important, we've done some of this education, is that simple hand washing. This is an enveloped virus like SARS-CoV-2. So, hand washing and taking some of those simple hygiene measures it can be really helpful in preventing transmission, and how you're interacting with your toddler. Abstain from eating after them or sharing utensils and things like that, because those toddlers going to preschool are the best vectors.
So women who are CMV negative and then seroconvert during pregnancy, obviously that's considered a risk. If they're CMV positive, like most people seem to be in various serosurveys, is that also a concern because of the potential for reinfection?
Yeah, the problem, so most women are CMV positive, 70% of the people in this country are CMV positive, but in other countries, it can be as many as 99%. So it's very hard to find people that are seronegative, actually. But there have been some studies done in Brazil where there's like 95% seropositivity, and Brazil actually has some of the highest incidences of congenital infection. So that maternal immunity is not necessarily protective, but it is thought to be perhaps better than being completely naive and then encountering the virus during a pregnancy.
So the issue is that if woman is infected, the virus will cross the placenta and infect fetal tissues, right?
Yes, exactly.
But in order to do that, the virus has to be active and not latent, or is it, we're going to get to the whole latency thing, but one of the keys to this virus, like any herpes virus, is that it goes latent and it's just hiding.
Right.
Is that a major factor in determining whether it goes and has access to the placenta?
Yeah, so probably. I mean, I think with herpes virus latency, the dogma has been sort of this very quiescent, latent state where the virus is completely silent. And I think we're really, as a field, shaking that up right now. And there's been some fantastic work done, for example, in herpes simplex, which has herpes simplex 1 has the most classically defined latency in that people are infected, they may get a cold sore, the virus will establish latency, and then when it reactivates, the cold sore will appear in the exact place where they've had it previously. And some people, though, will be infected with HSV and never get a cold sore. Other people are infected and will have a cold sore every month. Other people, only a few in their life. And so it's very interesting, the dynamic that dictates that. But what people have done, scientists have done, is looked longitudinally over the course of months, having people swab like every 6 hours during a day, and they can detect herpes simplex virus shedding in like 28 of 30 days, even though there's no symptoms. And so we don't think these herpes viruses are nearly as quiet as we think they are. Just because you don't have clinical symptoms doesn't mean the virus is completely latent. I think it's sort of an important point. Of course, in the lab here, we study latency at the level of the individual infected cell, where you can really measure this very molecularly. But in an organism, where CMV, much more so than HSV, it will infect any type of cell in the body. So we're carrying a lot of CMV, and at any given time, you're probably having cells that are kicking off reactivation events, being cleared by the immune response so that you don't ever see that clinically or symptomatically. And we don't think probably latency in an organism is nearly so quiet as we once thought it probably was.
Okay.
So I guess how would you define latency then?
Oh, well, I mean, I think there's, clinical latency has nice definitions, right? It's basically the persistence of the virus in the absence of any clinical symptoms or disease. And so most of the world is latently infected with CMV. At the cellular level, the molecular level, we are really looking for an infection where the viral genome is being maintained. Viral gene expression is quieted. It's not completely restricted or silenced, and you're not making progeny virus. And so that would be true latency. And these viruses are, I think, much more noisy than we've given them credit for. We just, until the recent last decade or two, we really haven't had the tools sensitive enough to detect viral activity in the latent context, especially within an organism.
But if you look for infectious virus, but you said earlier that it takes years of infection before you really get into some kind of latency. So if you have people tracked individuals for extended periods of time and look for infectious virus production, and what do you see?
Not so much with CMV as been done with HSV, which is a little bit easier. CMV, there have been some, and you can detect CMV, I think, far more often in urine than you might expect in a latently infected individual. But those studies, I think, really need to be done in a much more in-depth way, especially now that we have some really great, abilities to get to, super sensitive detection methods.
So what do you think is happening? Do you think there are bursts of infectious virus production?
We do. We think there are micro reactivations that are kicking off all the time, but are being limited by the immune response, and those cells get cleared. And so they never rise to anything that you would ever notice clinically. Okay. And of course, most people, even when they're initially infected with CMV, will not have any disease. So this virus is really... It is very stealthy, and it's
well adapted, right? It's well
adapted.
Now, with other herpes viruses, especially HSV, which has been extensively studied, we talk about latency in the trigeminal ganglion or latency in neurons. Where does CMV hide?
Well, where we have studied it is in hematopoietic cells, and that's because from a human being, you can get hematopoietic cells relatively easily. There could be other reservoirs. Those can't really be excluded at this point. There have not been any really comprehensive studies. We did partner with a lab at Columbia University near you, Vincent, with Donna Farber, and we were trying to basically screen tissues from people who had donated their bodies for science, and we would take tissues from all organs, and we were looking for CMV. The problem is the virus is maintained at a very, very low level, and so these, the detection, even with the sensitive techniques that we have, is very, very difficult in a person who doesn't have any sort of disease. Or, for example, it's easy to detect CMV in a transplant patient, very hard to detect CMV in someone who is otherwise healthy. So it's really challenging.
So you said virtually every cell can be infected? So epithelial cells, respiratory, gastrointestinal, endothelium, yes. All of that? All of it. And that's, CMV is very restricted in its species tropism. So the human virus will only infect human cells. But within the human, any cell type is pretty susceptible. So the virus uses multiple receptors. So there are other CMVs that infect other species, other yes. animals? Yeah.
Do we know what limits the viruses to their specific species?
Yeah, there's been a little bit of work done on that, but it's basically, the viruses are so co-evolved with their species that, the viral program just doesn't quite kick off. Immediate early genes don't quite function the way they should function in an animal where the virus has not evolved. And so, for example, I study a block of genes in the human virus that is present in the chimpanzee CMV. But when you get to rhesus, the region of genes is there. There's a big space that looks like it should be these genes because it's flanked by genes that are highly conserved, but there's no clear orthologs to the genes that I study in the rhesus virus. And then in the murine virus and other rodent viruses like guinea pig or rat, these genes actually don't even exist. And so the genes we're studying are really very specific to an adaptation that has happened in higher order primates, and most specifically chimpanzees and humans.
Now, given the extent of this adaptation, this must have been the original ancestor of CMV must have come into us very, very early, right?
It's estimated that it predates the invertebrate split in evolution.
Okay.
CMV is part of us. It's basically like an endogenous retrovirus.
And is it pathogenic in other species? Like more pathogenic than it is in humans?
No.
Okay.
So do people study these I guess people study murine CMVs, right?
Yeah, the murine virus is quite different from the human. There's only about 70 genes that have any homology, and it's a much smaller virus, replicates much more quickly, has really kind of been reclassified. But yes, the murine virus has been critically important, and especially understanding immune responses and pathogenesis.
So you said that it's a problem in immunocompromised people. So I think one of the issues is if you have an organ transplant, this could be a problem, right?
Yes.
So you get immunosuppressed, and they have to watch to make sure that it's not getting out of control, correct? And
they monitor very carefully in any transplant patients. And in fact, antivirals are generally given preemptively if the recipient is positive for CMV.
Got it. Okay. So let's talk about what is regulating latency and reactivation. It's one of your main areas of interest, right?
Yes, so very complicated. But the virus is going to enter the cell, and we don't really know exactly what happens. But we think that even in a cell that's non-permissive for replication, where the virus will establish latency, there's some gene expression that is kicked off. Nobody yet knows how important that is, but eventually that gene expression will get largely silenced. I mean, there'll always be some low-level gene expression happening, especially of these genes that we think are important to a latent infection. And then that virus infection will sort of stay in this quiescent state until something stimulates the cell that is basically feeding it an input that's telling the virus that it's time to get out for whatever reason. And so we're very interested in what are those cues and how does the virus sense those cues. And so I think one of the overarching themes that has really come out of our work is that mostly the virus is using the natural cell biology that is there. So every single host cell factor, for example, that we have found that it is important for latency or reactivation, and this can be things like homeostatic signaling through the epidermal growth factor receptor, AKT, PI3 kinase, that sort of signaling is feeding inputs into the virus. And so how is that happening? The virus has basically evolved to be able to sense this because it has all littered throughout its genome host transcription factor binding sites. So when something changes in the host signaling, that's then going to change the viral gene expression that's happening. And every single host factor that we have discovered that has been important for CMV latency and reactivation is regulated by cellular differentiation, which is one of the major reactivation stimuluses that we know. So hematopoietic cells, where the virus establishes latency, they make up all the cells of your circulating blood system. We study the virus in progenitor cells. So these are undifferentiated cells that have a very specific transcriptional program. And when you induce differentiation for those progenitor cells to become myeloid cells like macrophages, monocytes, you'll get a very dramatic shift in the transcriptional program. And so host transcription factors that are important for latency will be highly expressed in these undifferentiated cells, such that when the virus encounters that cell, it really has a transcriptional milieu that is set up to drive a latent infection. And then that transcriptional program and those specific transcription factors will completely change as soon as the cells differentiate and they go away. And then you'll get other things that are actually really important for stimulating viral genes that are important for the reactivation event.
So the virus literally knows us better than we know ourselves.
Yes, that's what I'm always telling students. It's a master of our cell biology.
And are these the types of factors that are different between the human and chimp virus versus the rhesus virus? So in fact, they know us and know how we're different than those other organisms?
Yeah, potentially. No one has really looked. The rhesus virus has been used exclusively because it is, you are using primates. That virus is pretty exclusively used for understanding the congenital infection, where it's been a really important model, and also understanding pathogenesis and immune responses and how CMV could be used as a vector, for example. So we don't really know a lot about how these sorts of things we're finding translates into the chimpanzee or rhesus virus, at least at this point.
So when I teach herpes viruses, I start with herpes simplex, which makes perfect sense at the level I understand it, right? Virus gets into peripheral neurons and it's quiet and there's no viral proteins made, so there's no immune response that's going to happen. Then periodically it gets reactivated so it can get another host, right? Because if you just stay in the initial host, that's not a good end game.
Not a good end game.
All right, so that's beautiful logic, but CMV always confuses me. And as you've just said, any little perturbation of a transcriptional program is going to reactivate it. So, taking the herpes simplex paradigm, how do you view CMV? What's the function of this responsiveness, I guess?
So a lot of work in the last, decade or less has shown that even a herpes simplex infection is not as quiet as we think. And there are probably proteins being made at a low level. CMV is probably a little messier. It's a much bigger virus, and it doesn't seem to be as quiet or to have as well of a controlled latency. I think we could probably invite 2 or 3 other herpes virologists on and debate that all day long. So it's just, I think it's a little bit more complex in that regard. And because it's infecting multiple cell types, I mean, it really is integrating a lot of different information and has to know how to respond as things change. And that's been sort of one of the complexities, I think, but just such an intriguing area to study. So, for example, one area that we're just getting ready to publish a paper on is where one of the genes that we study is absolutely required for reactivation. If you don't have it, the virus cannot move out and productively replicate again. It's being controlled by a host E3 ligase that is controlling things involved in cholesterol homeostasis. And so it's like the virus has somehow evolved to use this pathway and maybe be sensitive to changes in cholesterol, for example, in a host cell to know time to reactivate by having a critical determinant be controlled by a factor that's essential to controlling cholesterol homeostasis. And so I just think that, it's just incredibly complex, but incredibly elegant.
So would it be accurate to think of this as the virus can sense all these many, many different things within the cell, and maybe when some of them tickle it, it turns on partway, and others, so that might explain why it's constantly in this sort of fluttery state of partial reactivation?
Yeah, I think you're hitting on something really important there. I think that it is about gradients and thresholds, and that the virus has to cross a series of thresholds to productively replicate. It's not going to be just one host factor. It's not going to be one viral factor. I mean, it's really a very layered process, I think, to where the virus can kind of come up as some tickled response, but then decide maybe not to go all the way. And those are of course really hard things to parse out in the lab, but that's exactly kind of where we are trying to do that.
So less a toggle switch and more a rheostat.
Exactly,
right.
Which comes down to the sort of idea of it's not just an on-off state.
And I think that what strikes me from hearing you talk about this is I could imagine that rheostat is one thing in, say, herpes simplex virus, but in CMV that has to be sensing that range of things in so many cell types that are going to each be having so many things going on in them. It makes sort of further complexity, and also means that the virus is just responding to way more signals, or even measuring way more signals.
Right. Well, and herpes simplex, of course, is establishing latency in neurons.
Right.
And so that's a terminally differentiated cell, whereas CMV establishes latency in cells that are still able to differentiate, which makes things a little bit more complicated. And then I should just clarify that while we think latency happens in hematopoietic cells, there are other cells where it's very clear, like fibroblasts, epithelial cells. They could have a low-level chronic smoldering infection in a organism for a while, but they may not ever have any sort of thing that would look like latency. So there's probably cells in the human that will only support sort of a productive infection, and then there's cells that will support a latent infection, and then of course there's the interaction with these cells, especially as they circulate, like hematopoietic cells that are interacting with endothelial cells in the vasculature, picking up virus or seeding virus, and these sites where we think that the virus can actually replicate at low levels for a very long time.
Are there long-lived precursor cells which are latently infected? Hematopoietic precursor cells?
And that's how we came to study hematopoietic progenitor cells, is that really all the early literature, they could detect viral genomes as far back as the CD34 hematopoietic progenitor cell, which can give rise to really every cell in the circulation.
So do you view the constant reactivation as the function is to make enough virus to infect a new host?
Yes.
Sort of like herpes simplex paradigm, right?
Mm-hmm.
And
yes,
so
exactly.
When we don't have reactivation, although I'm getting the sense that we may not have a clear barrier between the two, when we're in a latent phase, when there's very little infectious virus, maybe not enough to infect another host, what's the immune response doing there? Is this below the radar?
I don't know that any sort of virus replicating is below the radar, but the one important thing to consider with cytomegalovirus is that they are the masters of immune evasion. They encode about 7 proteins just to escape MHC class 1 presentation. And this has been shown to be incredibly important for the persistent infection. And the rhesus virus was used to do that. So it's not exactly clear, and it's a big question for those of us that carry CMV for our lifetimes, and we're kicking off all these micro reactivations. What is happening to our immune response? So we know as people age, by the time they are reaching their 80s, up to 30% of their circulating CD4 and CD8 T cells can be specific to CMV.
That's amazing.
We are devoting a huge amount of immune power to controlling this virus that people think of as not causing disease.
And it's the MHC1 antagonism is to prevent T cells from killing CMV infected cells, right?
Yes.
MHC1. I always can never remember 1 and 2. 1 is the one on all cells, right?
Yes, and cytomegalovirus not only dramatically alters MHC class 1 presentation to protect the infected cell from T cell killing, it also alters MHC class 2 presentation, innate immunity. It really is quite skilled at completely thwarting our immune protection. And so, Klaus Frew and Louis Picker at OHSU have done just gorgeous work studying this in rhesus macaques, where they have shown, and it has given rise to the idea that you could use CMV as a vector, a vaccine vector, because they have shown that you can continuously infect these CMV positive animals over and over again with CMV and get brand new immune responses as if the organism has not seen that virus before.
So in a fully immune animal where you have T cells and antibodies, you can still get infected.
Yes, and a woman who is infected with B cells and has B cells and T cells to the virus can still pass it to a child through the placenta.
So do we understand why that happens?
No, I mean, it is a huge area of research, is people really trying. I mean, Klaus Frew and Louis Picker have spent a lot of time studying these immune evasion genes that are really important. And then, Sally Permar is studying how the congenital infection really happens, and what would it take to get a protective immune response that would keep it from crossing the placenta. But this virus is exquisite in its ability of escaping our immunity.
Fortunately, it doesn't cause disease most of the time, right?
Right. I mean, it's not that it's fully escaping our immune system. I mean, I honestly think it's using it, right? It's using it to sort of keep its level at just that sweet spot where it's not causing disease, but it's able to persist and spread.
I would call it a Goldilocks spot, right?
Goldilocks spot. That's a fantastic analogy.
The favorite immunologist analogy.
Yeah.
That's right.
Yeah. So I guess, correct me if I'm wrong in how I'm understanding this as you're talking about it. The idea is that the virus is sort of evading the immune response enough when you are infected that you never make a fully protective response. So it's not like maybe somehow you couldn't figure out a way to make a protective response somehow, but because of the immune evasion that's coming along with infection, you're not actually getting that to happen. Is that?
It's not because there's so little virus there or something like that, but I actually think this virus is actively shaping our immune response to it. So it's shaping it into something that it can live with.
This is what happens when a virus has been around a long time, and maybe SARS-CoV-2 in a million years will be similar, right? It doesn't
just a million.
Yeah, right. Well, that probably doesn't take. Well, she said it. The split between but there's that too.
More quickly.
But I do think that if you're with people a long time, then the guy at Sloan Kettering, the writer, what was his name, Alan?
Lewis Thomas.
Lewis Thomas. Yeah. He said there's this negotiation between pathogen and host, and most of the time it's okay, but sometimes one pushes a little too hard, and then you have disease. But if you've been around a long time, you get to this level where everybody's happy. right? The virus gets a place to live or to reproduce, and the host gets to live without disease. And the other thing that strikes me is that there's no inflammation really, right? So the regulation of the immune response by the virus is also keeping that low, which is really good because we know inflammation can cause disease on its own.
Exactly.
Right. What I wanted to talk about the genome a bit because you do some work on genome stability. What's the state of the genome in a latently infected cell?
So it doesn't integrate. A lot of people think about viruses that persist for a long period of time, like HIV, are going to integrate. It does not integrate. It exists in our nucleus episomally, and we don't even know of a real mechanism for tethering the virus to the genome. And so those are big open questions about how this genome is maintained. And because this virus is so large, I mean, we kind of take it for granted how much control it has over things. We think it has a lot. So, the virus encodes its own DNA polymerase and processivity factor. It has a lot of genes for replicating its genome. But we recently looked at, which is why I love academic science, is this student who had graduated and was kind of looking for a new project to do while he waited for his wife to graduate from medical school, appeared in my office door one day and he said, CMV encodes a polymerase, right? I'm like, yes, you've been in this lab for 6 years. How can you not know this? Anyway, just hang with me for a minute. How can it replicate its genome, which is a pretty complex genome? There's lots of repeat sequences. There's GC-rich regions. How can it replicate its genome with a single B family polymerase, which is like our epsilon or delta polymerases that do the bulk of our DNA synthesis, if a mammalian cell requires 14 polymerases? And I'm like, I have no idea. He completely set me back on my heels. And he's like, well, can I ask? And I'm like, yeah, go ask. And it wasn't completely out of the blue. I mean, we were studying a viral gene that we knew had an interaction that was able to modulate a DNA damage response, which was most important for this thing called translesion synthesis. And so in our genomes, when an error has occurred, like a thymidine dimer or something induced by UV irradiation, our polymerase will be moving along, encounter that, and stall because of the geometry of it. The polymerase can't move past it to synthesize that DNA. At that point, another polymerase has to come in that's going to have a much bigger binding pocket, and it's going to be error prone. It's not going to have proofreading activities, but it's going to allow synthesis over that weird geometry so that you don't have a fork stall and a collapse, which is the worst thing I think that can happen in DNA synthesis. And so you may end up with a mutation there, but at least you've gotten over that and it can be repaired later. So he had evidence that one of the viral genes was playing with this pathway. And so that's why he wanted to look at these, what are called translesion synthesis polymerases, because they're very important to this DNA repair. And so he knocked them down and he showed that they were important, had effects on virus replication, fairly modest effects on virus replication. So we're like, well, why does this matter? So we need to know if this is actually doing something to the viral genome. And so we sequenced viral genomes that the virus had only been passaged through cells that lacked these TLS polymerases one time. But he was leaving, his wife was graduating. We would have liked to have done like serial passages, but we thought, well, let's just see what we get. And the viral genomes are completely messed up with rearrangements, novel junctions that are formed without these host polymerases. So in this virus that we thought was so exquisitely independent in its ability to replicate its genome, it is actually using these host polymerases, which we've never thought of herpes viruses needing.
So I'm trying really hard right now to not totally geek out based on Vincent's previous inflammation point and the fact that I think a lot about DNA sensing. But I noticed one thing that you mentioned that I'm now starting to think a little bit about, which is that you mentioned we aren't sure about how the genome is tethered to the host genome. Can you talk about what you mean by that tethering?
Yeah, so for example, human papillomavirus is another DNA virus that can persist for quite a while, and it can be tethered to the host chromosome by a cellular protein linkage, basically, that will tether it to host chromosomes, so that then when that cell divides, the viral genome can get synthesized and then can segregate right along with the host cell. CMV doesn't have that. And so the problem with that is that as the cell divides, you could just get dilution of the genome over time. And we know to some extent that does happen. But if it can't tether at all, it could be completely lost. And then maintaining that lifelong latent infection that we all have would require a constant reseeding of a reservoir. And these are just open questions, like we don't know.
So the virus is in a epithelial cell, and when the cell divides, you don't know how it partitions to the two daughter cells, right? We have no idea.
Right. In an epithelial cell, the virus replicates to a level that there'd be lots of genomes there.
Okay.
But the other thing that happens, if the virus is productively replicating in a cell, that cell's no longer, it's not going to divide again. It's only in latency where the cells are infected and continuing to proliferate.
So in a latently infected cell, there's no DNA replication?
That's a good
not
question.
binary, Vincent.
That would definitely not be part of the classical definition. You wouldn't have DNA replication happening, but I think we just don't really know.
Because with herpes, there's no DNA replication, right?
Right.
Herpes simplex. But we don't know.
But that's in a cell that's terminally differentiated. So once those genomes are there, they can persist.
But you could also imagine that if there was that possible loss in some cells when the cell divides, that might be part of the reason why there's so much sort of productive viral infection and sort of the noisy virus, because cells keep having to get reinfected.
Yes.
Back to DNA replication, you said no DNA replication is not part of the definition of latency for CMV. So there may be some DNA replication to give enough copies so that when the cell divides, they simply get a little bit of viral DNA, right?
Right. We do know there is something very special about DNA replication. And when the virus is committed to DNA replication, we think that you get very strong signals important for reactivation. Mm-hmm. So there's something that's very tightly tied in with DNA replication, which I find completely delicious, but we don't completely understand it right now.
I never heard anyone say delicious in terms of
the context of viral replication, right?
That's very good. I like that. Delicious. You need new ways of describing things that really fascinate you, right?
Yes.
Delicious is a good one. So that just reminded me when a lot of people study sensing of DNA using CMV, mm-hmm. right? But
yes.
That's not your area. I know that. But as the DNA is coming in, it's already sensed on the way in. But you're saying that the virus encodes proteins that suppresses that, right?
Yes, it definitely does.
So the cell is not responding by making interferons and so forth. Okay. Wow.
Of course, a lot of what these viruses do, we think, for latency is they're kind of using some of these restrictive pathways in the cell, like the intrinsic or innate immune responses or DNA sensing could be commandeered by this virus to sort of help it into this latent state. It's using that as sort of a suppression to impose upon itself.
That's delicious, yeah.
I'm trying not to geek out here.
Yeah.
That's delicious. I'm not going to stop saying delicious for a while. So I want people to understand this virus. How many envelope glycoproteins are there in CMV? Do you know of it?
Oh, my goodness. Envelope glycoprotein. Well, there's GO, GL, GH, GE are some of the major ones. There are a number of envelope glycoproteins, quite a number. And they form different complexes on the surface of the virion that's required for entry into all these different cell types using different receptors.
Right, that's the key because you have to infect so many cells. Yeah.
Yes.
I want people to understand that not all viruses just have a spike, right?
No.
Because people have been influenced in the past couple of years by a certain SARS-CoV-2 with a spike, right?
There's nothing quite so simple and elegant as spike.
Yeah. So do we know what receptors are needed for this virus to get into cells? Yes.
Yeah, so PDGFR is a really important entry receptor, so that's pretty ubiquitous. EGFR has been proposed as an entry receptor or co-receptor. Neuropilin 2, I think, is an entry receptor. So there have been a number that have been described over the past couple of decades, and there's a lot of different co-receptors, I think, that are involved in different cell types. But that's that's part of the secret as to how you get into so many different cell types.
And is anyone interested in a vaccine for CMV?
Oh, yes.
Yeah?
Yeah, so that's what Moderna was working on CMV prior to the pandemic. And when the COVID pandemic hit, they just switched their gears. But, the complexity of a vaccine for CMV comes from the fact that this immune response to CMV is so odd, and the fact that you can't get this sort of neutralizing antibodies, and the antibodies we make to CMV infection don't protect necessarily, from the virus crossing the placenta. So a vaccine in CMV is a really challenging thing to approach. Where CMV, the work that was put into understanding CMV and making the potential for making a vaccine has really kind of converted to now using CMV as a vector, because you could imagine like a pharmacy wall of all these CMV vectors to vaccinate people against any cancer or other virus that you can think of. And so there's those two arms, I think, of the research and how this will impact humans going forward.
You could just take unmodified CMV and use it as a vaccine vector? Do you have to do something to...
No, you would want to add. So the work that's been done in rhesus is adding the SIV GAG protein to the CMV as a vaccine for SIV, or in humans would be HIV.
Yep. I remember that paper out of Picker's lab where they did that, and they had like all these non-human primates, and like one or two got infected, and they don't know why, right? In the face of this. That's the big problem. So
they're starting to know why now.
We should actually have some of them on TWiV too. That would be interesting.
Yeah, they are. That work is it's constantly mind-blowing, delicious.
But if you wanted to make a vaccine, what was Moderna using? Do you know? Were they using glycoproteins or what? Because it seems to me like you wouldn't know which glycoprotein to even use, right?
Yeah, they are. There is a lot of focus on glycoproteins. I don't really know what the vaccine plans are. That's all sort of proprietary, above my pay grade.
And you'd have to give it at birth, right? I mean,
or close to.
Yes. Right? Yes, this would, or someone who happens to be seronegative, especially say maybe prior to a pregnancy or something like that.
But we know for RS virus, we have to do it close after birth also, right? So it's not a insurmountable issue. But so you said something I just want to go back to. So if you take serum from person who's infected, it doesn't neutralize virus infection, say, in a cell culture?
Yeah, so I mean, I have very high titers of circulating CMV antibodies, and so when I entered Tom's lab, they bled me right there at the lab bench, and it was completely worthless at neutralizing infection in a culture dish.
Wow.
Of at least a lab strain. So, but yeah, this is a problem, and so they have used actually serum antibodies collected from CMV positive people called cytogam as a way to try to neutralize infection, especially if a woman has seroconverted and has an active CMV infection and is pregnant. I actually don't know the efficacy of those. I think we'd hear about it a lot more if it was thought to be terribly effective.
It may not be that neutralization is needed for protection. It could be an FC-mediated function,
Oh, sure. And this virus is incredibly cell-associated. You don't get a lot of viremia. I mean, it moves cell to cell, and that makes it, much harder for antibodies.
Yeah, so we don't even really know the correlates of immunity, right?
I mean, I'm not a viral immunologist. I would never pretend to be one, but no, I don't think we fully know correlates of immunity.
Doesn't sound like it, right? I think, Alan, you could say that for almost every virus.
Well, but I mean, we have some, like poliovirus, if somebody's got antibodies against it, they're pretty well protected against infection. And SARS-CoV-2, you're pretty well protected against major infection.
We do know that T cells are incredibly important, and NK cells are equally important. So those things are critical for CMV. The antibodies are a much harder bag.
But Alan, even in the face of variants of SARS-CoV-2 that aren't neutralized, you're still protected from severe disease. And, it may be T cells, it may be non-neutralizing functions, who knows? So you have to be really careful. I want to switch gears to your other activities, if that's okay with you. Have we had a good conversation about CMV, you think?
I think we have. We've touched on a lot of high points.
All right. You published a paper called Virology in Peril and the Greater Risk to Science. So this is open access, I'm pretty sure, somewhere. So it's MBio and MSphere and JV. So,
and JVI, Journal Virology.
So you're the editor-in-chief of JVI, is that correct?
Co-editor-in-chief with Stacy Schultz.
Stacy Schultz. And you're also the president of ASV, right?
I just ended my term as president, so now I'm past president.
Okay. Congratulations. Yes. What is Virology in Peril about?
The SARS-CoV-2 pandemic, it was an unprecedented time in science where even though people didn't necessarily all come together around this pandemic and the way that we were going to approach it, scientists really rallied at all levels, and unprecedented speed to the production of very good vaccines, antivirals, monoclonal antibody therapies. We learned a lot about the pathogenesis of the virus. We were able to treat it. Less people were dying. This was truly remarkable. But amidst all of that, somehow, we got this really anti-science, anti-virology rhetoric coming up. Dismissal of vaccines, which has always been around, of course, but amplified with SARS-CoV-2 and attacks on science. And this has gotten really complicated in thinking about the origins of SARS-CoV-2 and all of that. And so the Science is in Peril is really about that piece of the attacks on science. And science really is our best defense against pandemic viruses. We've had 10 in the last 100 years. There have been, 3 in just the last 5 years? So, this is our best defense. It's our best defense against the climate crisis, which fuels the emergence of virus and the zoonoses that are happening to drive viral emergence. And so I think it's a point where a lot of us in science are really very worried about the climate for science, and there's, potentially very aggressive legislation to restrict science that would allow us to respond to pandemics and things like that.
And you're talking about the gain-of-function research restrictions that we've talked a bit about this on TWiV as well.
Yeah, and so gain-of-function, it's something that every scientist does. It's an experimental approach, right? Just engineering in a fluorescent molecule into a virus so that you can watch it move to cell to understand spread is gain-of-function. There's gain of function like that gives us things like cancer therapies, and there's gain of function that could enhance the pathogenesis of a virus or its pandemic potential. And these things, all of this science is incredibly highly regulated, and there's a lot of oversight for it. But this is where I think the argument's being fueled.
So who's the audience for this article? Are you talking to scientists primarily?
Yeah, it's all over the board. There needs to be conversations happening at every level. And so I've published a lot of things with a co-author, Jim Allwine. I participated in a Public Voices Fellowship run by the Op-Ed Project in 2018. And thought, okay, well, that was nice. I wrote an op-ed or two and thought, this is a great skill to have. And then the pandemic hit. And Jim Allwine was at the University of Pennsylvania and has retired to Tucson and was coming to my lab meetings every week just because, scientists don't really retire. And so one day after lab meeting, I just said, hey, Jim, can I talk to you? And he said, yeah. So he came back to my office and I said, I feel like I need to write about this. I need to write about, I need to use this ability to sort of write for the public and write about the SARS-CoV-2 pandemic and what's happening. I said, but I can't, I'm just so busy. Could we try writing something together? And he said, sure. And he excelled at this. And we had a great writing relationship. I don't know what
collaboration.
We co-wrote really well together. It was a great collaboration, and we wrote like 35 op-eds during the pandemic. And so those were all published in sort of The Hill, local newspapers, things like that. And now we're writing some things that we're publishing in MBio, Journal of Virology, and Sphere, and those are, yes, on some level for virologists. So kind of like try to have this conversation around what's happening, help virologists better educate the public, but also we can use these platforms still to push these sorts of stories and ideas out more into the general population.
I think MBio, MSphere, JVI, that's a scientist audience, right? People are just not, we could say it's open access on this podcast, and a few hundred people may go, look, but that's not the kind of impact that you're looking for. But what you want to do is tell the virologist, hey, you need to know this, and go out and talk about it, right? That's how I would do this, right?
And so over the holidays, Jim and I started writing something, and I was sort of like, I'm so busy, with manuscripts and lots of students that need lots of different things and grants. But I just, I looked at a draft that he had, and I'm like, yes, we absolutely need to do this. So we started shopping it around to colleagues and stuff, and now we have over 90 colleagues that want to sign on as co-authors. And so we're going to publish this, and it will be published in some of the ASM journals, but we're going to then work to make sure that we're getting that message out. But at least within an ASM journal, we can have these 90-plus co-authors that have signed on to this message. And I think that's going to stand as a really strong, cohesive statement that a lot of, very independent-minded people have been able to come around to say, yes, this is what we stand for.
And I would just like to put in a plug for the idea that in addition to this sort of, talking to the virologists and talking to the public through articles, there's a level of advocacy that's much smaller scale that can overall be a lot more effective if those 90 virologists are, dropping their kid off for school and talk to somebody who's hesitant to get vaccinated or whatever. I mean, that's an opportunity, and that's an opportunity that scientists really should be taking advantage of, I think.
No, and that's exactly right. And that's what we're trying to engender and empower in people, is that you really can make a difference. And it really does. The grassroots local level is as impactful as anything else we can do on a big scale.
I mean, I think in a lot of ways it makes a very large impact to each of those individual people because the stories are so convincing in a way that impersonal things may not be.
Well, in fact, these kinds of articles signed by 100 virologists, they will not get wide press attention. Right? Can you see the headline? Virologists agree on something, right? They're not going to publish it. But they will publish, 20 virologists who say we need to investigate the origin of SARS-CoV-2, right? That gets a headline because it's controversial. So it's unfortunate that it doesn't do better. But, as we have seen many times, headlines are all about news, right?
Yeah, the news is what's new, and if you're looking at it, that's what you're paying attention to. So it catches your attention. But it's important to remember that people don't form their opinions just from, or even primarily from, the news. If that were so, journalists would have a lot more power than we do.
Right.
Well,
and the conspiracy theories come from a small segment, and they're just being amplified so effectively. And so this is at least our attempt to say that a whole bunch of people with a whole bunch of expertise in this area disagree.
Unfortunately, as you know, expertise is undervalued now. There's a very good book called The Death of Expertise. I don't know if you've read it, but if you haven't, you should read it. Who's the author? Let's see. Death of Expertise by Thomas Nichols, Felicia. Okay, I would read it because that's what we're going through. in the world, and he identifies the source of it. There are multiple sources. It's not just the internet, but people have disdain for someone who says, yeah, I've been doing this for 40 years. I'm an expert. And they say, I don't care. I could look it up in 10 minutes and be an expert, right? That sort of thing. So it's very difficult.
And that's really sad. I mean, that's very different from, the period where I grew up, where we really revered people with expertise that had devoted their lives to understanding something. And it's something that I think we do. We do have to understand the origins of this and how do we counteract that. So I will look at that. I will get that book.
It's a very good book. You'll really enjoy it, and you'll feel bad at the end because, part of the problem is today's college/university. We're not doing students any favors by being nice to them, right?
Right.
We're not teaching them critical thinking and so forth. So it's a problem that's not going to be solved overnight. But I think this is a great thing to do, and the more virologists you get to sign on, the better. And virologists will read it and go, yeah, I should do this. This gives me some ammunition to do it. And hearing you talk about scientists responding, so I like to think that here on TWiV we responded too, right? We used to do 3 episodes a week and get people information. So we didn't make vaccines, but we explained them to people.
We made vaccines really against misinformation.
It's true.
That's it.
No, it's very important. TWiV is critically important. I know so many people who what they know about virology is from TWiV. So this is fantastic. And these are people that are not scientists, right? They just enjoy TWiV.
Now, many people write and say, we're not scientists and we don't understand it all, but we get what you're saying and we appreciate. So you have to, you said things today that a lot of people are not going to understand, right? EGF and PDGF, they're not going to know what it is, but they get the big picture, and I think that's really important there. So,
and if nothing else, they get to see how excited we all are about the delicious science and discovery.
That's true. They do remark on how excited we get, and we go down rabbit holes, and we talk about what could be possible and how we're wrong. They like to hear all of that because that's what we do as scientists, right? Most people don't know that. So the more they learn about us, I think the better. And turns out we're people too, and we like some of the things that they do, right?
Yeah.
Yes.
All right, Felicia Goodrum, University of Arizona. Thank you so much for joining us.
Thank you. It was fantastic to be here with you. Really
Great
fun.
having you.
It was delicious.
It was delicious.
It was delicious.
All right.
All we need is a good glass of wine.
Delicious. That's a new one.
Yeah.
It's good. Usually something you reserve for food or drink, right? It's a delicious experiment. Well, I'm glad to see that. I tell my students, we don't understand this about CMV, and that was right.
Yeah, we don't.
We don't understand. It's kind of messy. It's a little sloppy. So, but maybe that herpes simplex is a little messier than I've been portraying it as well. Okay, let's do some email. I will let Alan take the first one because the second one seems to be addressed to Dr. Barker, so we'll let her take it. Go ahead, Alan.
All right. Emily writes, love the show. Thank you for sharing the science with the world. I can't even imagine how hard you all work to put out these podcasts. It's a piece of cake. How hard Vincent works. I get to hermit in my daily life in Idaho, but I'm flying across the country to NYC to visit my 80-plus-year-old parents. I wear a mask in public, and I will be wearing a well-fitted N95 or equivalent mask on the plane. If I am exposed and infected on the plane, how long after infection would I be considered contagious these days? I received my 4th dose of Pfizer a few weeks after the bivalent became available and have received the flu shot. Thank you, from Emily.
Well, I think you could be shedding within 3 to 4 days.
I think part of her question, though, is also how long, like when does it end?
Well, it depends who you talk to, right? The CDC says 10 days. I say less, but nobody should care about what I say. I would say 7 days is probably enough. 10 is being a little more cautious because, it's not binary.
Right.
There's
it's not binary.
There are tails on all of this, which means that there's going to be a whole range of people. So one day, another day, and so forth. So if you want to be certain, 10 days. But that's a problem if you're probably not coming to New York for 10 days. You don't want to, stay in your hotel room for 10 days or wherever. So you wear a mask.
Yeah.
Which it sounds like you're doing on the plane.
So you have 4 doses, and your papers have just come out showing that immunized people are less able to transmit. Okay, so it does blunt the transmission. It doesn't eliminate it. It blunts it. So, we already know that not everyone who's infected actually transmits, right? Only a small fraction, maybe 20% of the people are actually the big transmitters. So you're immune, you're good. Don't worry about it.
The other thing to bear in mind is your parents live in NYC, and they're going to get exposed. Probably not by you, but they live in the city, it's and this virus is everywhere, and it's not going away. So I wouldn't get too worked up about your potential for exposing them to it.
I think masking is a good idea. You're vaccinated. Have a plan. If your parents get infected, make sure they have a plan to get Paxlovid.
Yeah.
As early as possible.
Yes. Let's not wait and see, right?
No.
Let's see how you do.
No,
just get the Paxlovid.
No, they're in their 80s. They are by definition high risk. Therefore, if they should keep on top of this.
Test positive, Paxlovid. Okay. Brianne, can you take the next one?
Sure. Rona writes, Dear Dr. Barker, I love watching you and your background. I appreciate your calm backyard and how it changes with the weather. I think this is when I was at my parents' house earlier. It was atmospheric how the darkness came in towards the end of TWiV, episode 968. Speaking of the end of episode 968, it was exciting to hear that Vincent picked my pick recommendation regarding Columbia's radio station WKCR. I watched Dr. Barker's YouTube of her immunology college lectures. Thank you for making them available and for free. Something I hope to learn more about is the mechanism of asthma and allergies, as my children suffer from these ailments. Episode 968 was chock full of talk about ACE2 and how coronaviruses bind ACE2. I was interested to hear when Dr. Barker spoke about the usefulness of ACE2 and how it regulates certain necessary body functions. Something I was hoping you could touch upon is it true that asthmatics have fewer ACE2 receptors? If that is true, what do you make of the theory that asthmatics with COVID generally fare better than one would have thought? Could you discuss that theory that people with asthma have fewer ACE2 receptors, and this is the reason that people with asthma may not get severe complications from COVID as one would have thought? Thanks, Rona.
I wonder if Rona likes that her name is derived from coronavirus.
Yeah, I don't know. But I caught that as well.
Yes.
I got the Rona, right?
Yeah. I actually don't really know much about asthma and ACE2. But I would imagine that any details about how asthmatics fare with COVID could be related to ACE2 levels, but also so much of the other inflammatory and immune responses that are going on in asthma that it would be hard to pin it to just the one receptor, especially since I think that we can see a few different things going on with entry there.
I have this article I just found in 2021, and they say, asthmatics have are not high risk for severe COVID. And they say, this is because they have less ACE2. I don't know any data that can show that.
I don't know anything about this.
Oh, my gosh. I mean, how could you even do that experiment, right? I just don't. And maybe
you'd have
having
to
less
eliminate
ACE2.
all other variables and determine that it was just ACE2 driving their difference in infection. And no.
Yeah, very hard to do. Because we can't knock it down in them. We can't do that.
Well, and do we have a lot of asthmatic lung samples that we can actually do gene expression analysis?
You might be able to get some, but it's just going to be a correlation, right?
Yeah.
So I don't think you should assume that ACE2 is involved. I mean, ACE2 gets blamed for everything. It solves all problems or it makes all problems. Now, apparently the XB1.5 has higher affinity for ACE2. So now people are saying this is going to have implications for long COVID, which I don't even
I
understand
don't know how
where
you get
that's
to
coming.
that.
But don't extrapolate from ligand receptor interactions to pathogenesis. Okay, unless you have some experiments.
And this is
support
coming
it.
from somebody whose a significant chunk of his career involved ligand virus receptor interactions. So
Yes.
yeah, they're
someone who
important to
thinks
look at, but they're not a sole cause of things.
As someone who thinks about some events that happen between entry and pathogenesis, I can tell you there are a couple steps there.
Couple of steps, yeah.
So Alan, this has blown me away how, I studied virus receptors for so many years.
You were into them before they were hot, yeah.
And then people say, oh, there's a higher affinity. It must mean greater pathogenesis. Where do you get that from? If I had said that in my career, I would have been defunded.
Well, people want simple explanation. And by the way, Rona, we're riffing here. This is not any kind of a criticism of your question, which is an excellent question. And as Vincent has pointed out by pulling up a paper on PubMed, this is something scientists are interested in. But it's not going to be that simple. But people want it. We all want it to be that simple, because if it was, then you could, somehow target that process. But
yeah. So here's an example. This has nothing to do with Rona, but someone sent this to me on Instagram today. She said, please debunk this. It's a headline from the LA Times, and it quotes a virologist at USC talking about XB1 blah, blah. It's just the latest and greatest and most infectious variant. It's amazing to me that this virus keeps finding one more trick to make itself more infectious and more transmissible. Well, guess what, virologist at USC? It's not. You're wrong. Oh, my gosh. But that's the headline that they love, right?
Yeah.
That kind of headline. The most infectious, the most pathogenic, going to wipe out humanity.
If it bleeds, it
bleeds. Oh, my
bleeds.
gosh.
Yep.
So, Rona, it's a good question, but I think it is very difficult to get a relationship between ACE2 and disease severity. Chris writes, what a fun episode. This is Chiara Mingarelli. I have to say that your scientists are as fascinating as your science. Well, they're not our scientists, right? They're scientists. Astrophysics is a passion of mine. And so I would love you to produce This Week in Astrophysics with Chiara as host. I told her I'm ready when she's happy with her career and it's settled. Yeah, This Week in Gravity. Wouldn't that be cool?
That'd be great.
Because that's what she does.
Yeah.
TWiV.
It's only because of TWiV that I am now so enamored with viruses and microbiology writ large. Viruses are far more complicated than elementary particles. I remember one of your scientists talking about how simple viruses are compared to her field of bacteria. I agree with Chiara that there is no downside to knowing more about how the universe works. Thanks to Microbe TV, I get to explore to my heart's content. I don't know about that. Yeah, I mean, I agree, but it's like saying it doesn't hurt to take the vaccine, so just take it, right? I think we should know how if it works. But it's not the same thing, I know that. Pulsar clocks measure low-frequency gravity wave 100 nanometer distortions of space-time over decades, measuring the gravity wave background separately from the capture of individual wave signals. Yikes! And atomic clocks are now even better. They can measure the difference in gravity from 1 meter to the next. I would like to ask Chiara, what is a graviton? Well, I will send your email to Chiara. I don't know. You know what a graviton is, guys? I don't know.
I think it's a particle involved in gravity.
Yes, right.
Oh, okay. Brian Greene once described being captured by a black hole like being caught in a river. It is kind of like not being able to swim faster than the river and getting caught. Inevitably, one goes over the waterfall or event horizon of the black hole. There's no escape. Light doesn't bounce off the river anymore, but gets caught in its flow, and we can no longer see it, making it black.
And the metaphor breaks down.
The only direction is down into the center, the singularity, unless, of course, there is an Einstein-Rosen bridge wormhole. I don't know what that is. Do you guys know what that is?
My science fiction understanding of it is that it's an
connection from one
anastomosis
part
in the universe.
to the
Yeah,
another.
it just connects one space to another through a kind of a gravity hole.
Okay.
I'm sure Kiara would have a much better explanation of that.
It's the way you travel across the universe quickly in science fiction.
In science fiction, yes. Wormholes feature very prominently in sci-fi, but I don't know the physics of them or even that any of them would work like that.
Well, I will send.
They're very useful literary devices, though.
It's kind of like a Deus ex machina.
Yes, it's magic. It's sci-fi's version of one of sci-fi's versions of magic.
And it's a joy to have Angela join the family as host of TWiV and have her sister as today's guest. Thank you for your YouTube post. Fabulous Sunday morning. I just love following you in all things. Fondest regards, Chris, Toronto in the snow. P.S. I went to Carleton University in Ottawa too, but maybe before Kiara was born. You shouldn't follow me if I walk into a dicey place, okay? You can follow the podcast, but maybe some other things. No. All right, let's do some more, and we are back to Alan.
Sure. John writes, Doctors TWiV. While watching Svante Pääbo's Nobel lecture from a couple of days ago, I don't recall mention of a correlation between a Neanderthal gene and severe COVID on TWiV. But looking just now, I see that you did this in number 676. That was about 10 epitopes before I started following. Happy to hear you say Neanderthal, too, throughout, and not thal. Anyway, here it is, presented by the man himself. The COVID risk part starts at about 47 minutes and links to the Nobel Prize lecture. Best regards from Greater Braddock, where it was a typical glum November day, fully overcast and not much above freezing, except that with global warming. Except that with global warming, November is now coming in December. John. And where is Braddock?
Yeah, we say Greater Braddock is Michigan, I think.
Or Pennsylvania?
It's Greater.
I've got a Braddock, Pennsylvania that I just Googled up, which would give about that weather.
Pennsylvania, yeah. Usually he puts, but this one didn't have it. Okay. Brienne.
Paul writes, TWiV panel. Ever since I read about how important the folding of protein is, I have been amazed how mechanical the micro world seems. As I fumble to attach an HDMI cable to my phone, I can't help but try to visualize a virus trying to aim at a spike at a receptor. Of course, in my mind, this is happening in a gooey mess. My dumb question is, how mechanical is our microbiotic system? Does it seem that way to you folks? From New York City, Paul. I don't know that I've ever thought of using mechanical for this, but I think that the sort of biochemistry of how structures put together and how different shapes match is a sort of a key part of how all of this works. I definitely explained to one of my non-science friends that some of what we study is just how shapes fit together. It's just sort of like really advanced preschool. So in that way, yes, I can see exactly what you mean as you're watching that, thinking about that visualization of the virus and the receptor and kind of how they're trying to lock in to match each other. So in that way, it seems that way to me. I wouldn't have called it mechanical, but then again, I'm not a person who is particularly mechanically inclined.
So what is mechanical? When I think mechanical, I think of metal clinking together.
I think of gears and things.
I think, biology, I think of it could be construed as mechanical if you substituted meat for metal and mucus for oil. But that's not a really good, biology is a lot more wiggly. It's not as... it's tempting to look at it as machines interacting, and that would give us mechanistic. We talk about mechanistic understanding, like these are mechanisms at work, but there's a lot more leeway and interplay and statistical variation in what's going on.
And as Paul mentions, the gooey mess.
The gooey mess, yes.
However, we do talk about machines when we talk about.
We talk about molecular machines. And when you look at things under an electron microscope or in 3D cryo-EM or crystal structures, they can look very mechanical.
I mean, if you look at models of the respiratory chain in mitochondria, man, those are mechanical. I mean, so I don't know that you can't have mechanical at that level. Why not? Machines. It's just all made up anyway. What does it matter?
It's not all made up. It's
it's got to be consistent, right?
Yeah.
I don't have a problem with calling molecular machines mechanical, but we'll see. People will write in, I'm sure.
On the other hand, my experience with machines has been that they're often a lot less predictable than you would think based on the design.
True.
Well, maybe that's just you.
Yeah, it could be. No, I think that's a pretty general.
It's me too.
And I even understand a lot of them, but they don't always behave the way they should.
Yeah. Okay. Neither do people.
No.
So that shows that we're machines, right?
Right.
All right. Dave writes, "Hi there, TWiV team. Just saw a video by a doctor, ZDog MD, December 9th, where he was talking about viral interference as being one of the possible reasons why seasonal flu basically disappeared during the first year of COVID. Just wondering about your thoughts on this. You may have already covered this. Dave from wintry southern Alberta, Canada." So I think influenza disappeared because of isolations and masking and people staying home.
Yep.
Theaters were closed, sports were closed, schools were closed. That's the simplest. That's the, what is it? The
Occam's razor.
Occam's razor. I mean, yeah. Why do you have to talk about SARS-CoV-2 interfering with influenza right now? It could, right? So, yeah, sure. But the thing is, SARS-CoV-2 is a good inhibitor of interferon.
Exactly.
So, interference is often where one virus induces interferon and inhibits the other. So that's just one mechanism, of course. So I don't think so. And I don't think, ZDog can have his own ideas. That's fine.
But I think ZDog MD, if that's the only name we have.
No, he's a doctor at Stanford.
Oh, he's actually
he's okay.
Okay.
He's an MD, and he has a podcast.
Okay.
Apparently now everyone has a podcast.
Everyone has a podcast, I'm told, yes.
I've been trying to get on ZDog MD because he has everybody but a virologist on to talk about SARS-CoV-2. It seems weird, doesn't it?
It is weird. Yeah, no, I agree. Right. Mitigation seems like a more likely answer.
Yes.
And as a consequence, we have lower immunity to flu and RSV. And so now that we're back in the world,
everything's coming roaring back.
And you can see China is doing the same thing with SARS-CoV-2, right? Mm-hmm. They were locked down for a long time. And what does that mean? They don't get a lot of infections. And so when you let the people out, boom.
Right.
Well, and China's vaccine uptake was not very robust.
But supposedly most people got 2 doses and not 3, which you really need to be robust. Okay, yeah.
So but and then also, a lot of people just didn't get it or didn't get even 2
You know what the problem is in China? If you talk about 5%, that's like a lot of people.
Well, that's a huge number of people, but the percentages are.
Right. Go ahead, Brianne.
No, I was going to say I've been explaining some of the RSV information to people with the idea of spreading the curve. When we thought about COVID, we wanted to all stay home to spread the curve, to flatten the curve so that everybody didn't get sick at the same time. Okay, well, we also flattened the RSV and the influenza curve, and now we're all going out again, and we're we're not flattening it, we're smushing it back together. Yeah.
Some of the things that were said.
I know.
We are back to you, Brianne.
Okay. Barry writes, I was just listening to TWiV 869 about EBV being a possible etiologic agent for MS. I have 3 important points. I wanted to clear up a little misspeak about HSV1 and HSV2. Oh, I hope he was listening today. I think I heard Rich say that HSV1 causes cold sores and HSV2 causes genital sores. That's a long-standing misconception in the community. Both viruses cause both kinds of lesions. In fact, there are now a huge number of HSV1 genital infections being diagnosed more and more. Just see some of Anna Wald's work. One cannot rule out superinfection and recombination between the 2 viruses, which seems to be present in some patients. Regardless, the seroprevalence numbers were pretty good. HSV1, VZV, EBV, CMV, HHV6, and HHV7 are each at about 60 to 80% of the American population, reaching 100% in the developing world, and HSV2 and KSHV are much lower, yet sometimes as high as 50% in some groups. I was also taken by the idea of using an existing antiviral to treat MS. MS's constant remitting relapsing is very reminiscent of herpes infections that exit latency, then return. Each acute reactivation could very well be the stimulus for a new autoimmune flare-up. I've kept a PubMed Alive alert for years, waiting for someone to try to treat MS with acyclovir or its derivatives with standard daily suppressive therapy. EBV is susceptible to acyclovir, but its TK and polymerase are not as sensitive as those of the alpha herpes viruses. I have even seen a lot of literature suspecting HHV6 as being the culprit, which means acyclovir would be useless. One would need to use val or ganciclovir because you need to attack the UL97-related kinase. But as mentioned on TWiV 869, it doesn't seem that any clinician has gone out on a limb and tried any of them. Finally, there was speculation about immunizing against EBV and its relative uselessness. On the contrary, look at what we've been able to do with VZV, another childhood-acquired herpes virus. Yes, we've been using the attenuated OKA strain for decades, thereby causing a latent herpes infection of its own, but with the advent of Shingrix, one could easily imagine a subunit adjuvanted vaccine against EBV that could work just as well at preventing MS flare-ups, like Shingrix reduces the number of shingles flare-ups. I do wonder if it's only a matter of time before clinical trials are approved for the use of Shingrix in pediatric populations to replace OKA as the standard of care. Feel free to give me a shout out. I've known Brianne for a while now, since ASV a few years ago, and more recently from our Jeopardy appearances. And Vincent, since he gave a talk sponsored by my mentor, Wade Gibson, at Hopkins in the 90s. Although I left academia at the end of 2021, I'm still in the game at NIAID in extramurals as a scientific review officer in the AIDS review branch. And this is Barry, who is formerly from Towson and who I have met at ASV and who was also on Jeopardy this year.
Was he on with you?
No, he was on earlier in the summer. It was between the time where I had filmed and when my episode aired, when I couldn't tell anyone I was on.
Yeah.
Where I suddenly saw him and sort of was trying to communicate with him without sort of talking about the fact that I had also been on.
Cool. So this is very interesting. Shingrix could replace OKA as the chickenpox vaccine. That's what he's saying, right?
I think that sounds great.
Yeah. That's very interesting. Yeah, because OKA is an infectious attenuated virus.
Right.
Very interesting. Someone asked me that the other night, and I thought it wouldn't work, but why not? If it's in a trial,
I can't see why it wouldn't work, and I think that's a great idea to prevent that latent infection.
Same glycoprotein, yeah. I thought because, the OKA is preventing infection that initiates in the respiratory tract, right? Mm-hmm. And you could imagine that the correlates are different from when you're reactivating a latent infection in the ganglia. But maybe not. Maybe it works. Cool. Very cool. Did we skip you, Alan?
No, I think it's my turn, actually.
Okay, go ahead.
Okay, so that was from Barry. Joe writes, "Hi, TWiV team. I have been a big fan of TWiV and all the related podcasts since the start of COVID. You guys have been a wealth of knowledge and a calming voice throughout the pandemic. I'm a biochemistry grad student with no virology training, so I apologize if my question is a little naive or was answered in a previous episode of TWiV. Since the start of the SARS-CoV-2 pandemic, it seems like terms about viruses are being used very casually by the media." Yeah, we noticed that. For example, I am still confused on the difference between species, strain, and variant when referring to a virus. I'm sorry, Joe, I am not laughing at your question. I'm
laughing with you.
I am laughing along with you. Why is every new variant of SARS-CoV-2 a variant and not a strain? How many mutations or substitutions does a virus have to accrue to become a new species? I am further confused when we use the term subvariant. I understand what the media means by this term, but is there a formal scientific definition? It just seems like we are drawing lines in the sand on what to refer to each new version of SARS-CoV-2 as. Why is every lineage of Omicron a subvariant? At what point are we going to use a different Greek letter? Thanks for everything you do, and again, sorry if my question or questions is naive. best Joe. Joe, not naive at all. So I'll start with the bigger picture, the question of viral species. If you go back to, we've had Jens Kuhn on a few times, right?
Yeah, he did a taxonomy, didn't he?
So the taxonomy episode with Jens Kuhn is a good reference point. It's not a short episode, and it gets well into the weeds of taxonomy, but the question of what is a viral species is one that people have been arguing about for, a century and a half? And it's one that we're not likely to settle anytime soon. A very, very interesting question that is going to remain a question. In terms of
that episode was called Mistletoe on the Tree of Life.
Yes.
Isn't that great? I'll put a link in.
And then in terms of variants and strains, these, I've heard various definitions even within science about what's what, depending on what model system somebody's working in. But I'll let Vincent tackle more specific issues on that. I can deal with the press side of it, which is just, these terms are being thrown around casually because it's a way to generate news stories. I mean, you got to have something new. And I don't know why, I don't know at what point we say that something is the next actual variant as opposed to a subvariant of Omicron, or if we will ever say that. My personal theory that I've mentioned a couple of times is that this is really just all a conspiracy to deprive me of pie puns.
Yes,
because
exactly.
that is the next variant we should
good.
be getting. And some of these things probably could have counted as pie, and I want my pie.
You'll get it. I'm sure
Maybe
you'll
I'll
get
eventually get my pie.
Maybe it'll come out of China.
Maybe.
You know? So a variant is simply a virus with a different genome sequence. So if Alan and Brianne and I all have COVID and we isolate SARS-CoV-2 and there's a one base difference between each of ours, each of us has a variant. That's it. And a subvariant is it's a stupid term, really, because it's just another variant. But if you know the directionality that Alan's variant came from Brianne's, you could call it a subvariant. Okay, that's all that means.
And a strain is a variant with a unique biologic property.
A phenotypic difference.
Yes, a phenotypic
and
difference.
Jens wrote in an article, which is very funny, the designation of a species would have to be made by an international committee of experts. Good luck getting them to agree.
Because that actually is one of the things that he does.
Yes, that's right. So,
and you can already imagine what a phenotypic difference is also certainly up for debate.
Yes.
Yeah, I mean, how extensive should the difference be and all that? Yeah, it's very difficult. Why is every lineage of Omicron a subvariant? Well, if you look at the websites that post the phylogenetic trees of these isolates, you can see distinct branches, and those have not gone off the Omicron branch. You know, there's a big piece of the tree coming out of the trunk that's Omicron, and there's just little branches. There's not a whole new one coming off. You could look at it that way. And until that they're all subvariants. Hey, did you see that XB1.5 is a recombinant of two previous Omicron variants? Isn't that cool?
Yeah.
The first recombinant to be immune evasive. It's very cool. All right, so I'll put a link to TWiV with Jens, and also I did a video a long time ago defining variant and species and isolate, blah, blah, blah. Blah, 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 blah.
Blah, blah.
Okay, let's do one more. Maureen writes, let me write the end of the email here, so otherwise I forget. Hello to the good people of TWiV. Once again, an episode connected really well with my class. So Maureen is a professor up in, what's that college upstate?
It's in Rochester.
New Paltz.
Oh.
New Paltz.
Okay.
SUNY New Paltz, right?
Okay.
Yeah. Maureen used to be a student in our department, so I know her. I actually visited once. To help my virology students understand how scientists are learning about the origin of SARS-CoV-2, I shared the genome comparisons with Lao, I think Lao bat, Lao. Yeah, Lao. So I used to pronounce it Laos, right? But someone from Lao said it's Lao. Lao bat coronaviruses, and revisited the term zoonotic. I also discussed some tactics used by folks who spread conspiracy theories. I was so pleased to be able to follow up on that discussion by sharing Dan Wilson's Debunk the Funk YouTube channel. My students have a fun short writing assignment at the end of each class. One option is to make a connection between the lecture topic and a personal experience. A student who works in a hospital wrote about various SARS-CoV-2 origin stories her patients shared, including a Bigfoot origin theory. Her tongue-in-cheek question was, so this would be considered zoonotic? Yeah.
Yeah.
Different species, right?
Mm-hmm.
So what is a Bigfoot? What's the species?
I think
Should we
they're
look it
actually
up?
related to unicorns.
Oh, yeah.
And Yetis?
Yetis.
Also?
They're very closely related to Yetis, right?
Bigfoot, also known as Sasquatch, is a purported ape-like creature. Okay, so it's a different species. It would be a zoonosis.
It would be a zoonosis. We'd have to figure out if its CMV was
Right, yes.
similar to ours or not, like Felicia was working on.
All right, let's cease our email for now and move on to some picks of the week. And let's start with Brianne. What do you have for us today?
So I have a video made by someone named Acapella Science. We've looked at some of his videos before, where he takes songs and changes some of the words to make them about a science topic, and they're popular songs. So this is called Leukocyte. It is to a song by BTS called Dynamite. I was not terribly familiar with the song, but this Leukocyte video, it's about 3 minutes long, and it talks about a lot of different little parts of the immune system, particularly some parts of trafficking and what some different leukocytes do. It's quite catchy. You will have it in your head for a while after you watch it. And I really enjoyed it. Someone shared it with me on Twitter, so I thought I would share it with you all.
Very cool.
Yeah, this is cool. A lot of people are talking about this. Yep. I tried to get him to come on to TWiV.
Oh, really?
Yeah, but he just kind of said, what do you want me to do? I was like, well, come on and talk about what you do.
Yeah. What's your background? How did you learn all of the
how'd
immunology
you end
you
up
needed
doing
to do
this?
to make this video?
And then since you are a cappella, you could sing right here at the mic,
True.
right? Maybe I'll try again, because it would be fun to have these kind of
Or he's
people.
too big
Yeah.
a star. He's
Sorry.
got to talk to his people.
You got to press the flesh. Alan, what do you have for us?
I have, it's kind of a double pick. First item is a news story in the category of, well, it's about time. West Point, the United States Military Academy at West Point, is removing Confederate monuments from their campus. Okay, now, if you think about this for a moment, if you think about it objectively, why would there be Confederate monuments on the campus of West Point?
That's the other team.
Yeah. I mean, the whole thing with West Point is duty, honor, country. And what's the worst thing you could possibly do to betray the oath that all West Point cadets take? Well, it would be to take arms against your own country, which is exactly what the Confederacy was about. So I also have a pick of the week, which is a book that I read actually a little while ago, but wanted to go ahead and bring it up because this is timely. It's called Robert E. Lee and Me by Ty Seidul. Ty Seidul is a retired brigadier general from the Army, and he's a professor emeritus of history at West Point. So he had a long career in the Army, rose to brigadier general, then was professor of history at West Point, and wrote this book as his explanation behind the history of why West Point has Confederate monuments. which is enlightening, but also his own personal history as a Southerner, his own personal reckoning with the legacy of Robert E. Lee, who is hailed as something of a saint. And if you were brought up Southern, you probably have experienced this in at least some context. I certainly did. So this resonated on a lot of levels for me and was just a fascinating read about the structural racism that gave rise to these monuments, which were not put in right after the Civil War. In fact, the Civil War, the fact that Robert E. Lee was an alumnus of West Point was an enduring point of shame and horror, as it should be for the institution. And they had nothing to do with that until well into the 20th century when the academy started to become integrated. And then with each new step in racial integration of the academy, a new Confederate monument would pop up.
Wow.
So anyway.
Interesting. Good, nice connection. Yeah. It's cool. All right. I'm going to pick the works of Haruki Murakami, who is a Japanese writer who I've read for many years. I really like his work. I've been reading quite a bit of it lately on my Kindle app. So I just finished South of the Border, West of the Sun, which was very, very good. He's written many, many long pieces like Norwegian Wood, Kafka on the Shore, The Wind-Up Bird Chronicle. 1Q84 is a really good one. Very good. But he also has short story collections, which I hadn't read before, and I've read them in the past few months. And there are two collections that I've read. One is called Person Singular. Now, not all of his short stories are compelling. Some of them are just too weird, and I just can't get it. But, his stories are all about angst, and, they're translated from the Japanese, so they have a different feel about them. But the prose is very crisp and very beautifully described. So that's probably why I like him. But in this collection called Person Singular, there's one short story that stands above the rest, and it's called Confessions of a Shinagawa Monkey. So this guy goes to a hot spring. So he goes to a hotel that has hot springs, and he goes at night to the hot spring, and there's a monkey there who says, "May I help you, sir?" He speaks English. So it's a funny kind of situation. But he starts to talk to the monkey. And Murakami has little elements of biology in his stories. He seems to know just enough biology and genetics to
be
kind
surreal.
of make...
Yes.
Yeah.
And so the monkey said, I started to like human women. And he said, genetically that doesn't work. That was very funny. But then he said, I got to like human women so much that I couldn't deal with female monkeys. And this is a very interesting conversation between him and the Shinagawa monkey. And then there's another collection of short stories called Men Without Women, where it's various stories of people who have lost their partners to death or divorce or whatever. And the one that I found absolutely wonderful is Drive My Car. Right, this is a story about an actor who, for some reason, he has to be chauffeured back and forth to his acting jobs. I forgot what happened. And the chauffeur is this very interesting woman, and who doesn't talk to him at all for the first days, and then starts to talk to him. And he tells the story of his wife having an affair with one of his actor partners in the job. And it's a very interesting story. So, but also again the narration, the image that he portrays, the atmosphere. He's really good at that. And sometimes, so I like him very much. And many people do know him. He's a very well read author. But I wanted to recommend it.
Very cool.
Karen, we have two listener picks. Karen writes, I found this just published article thought-provoking and feel sure you will too. It's called On All Fours, Transient Laborers, The Threat of Movement and the Aftermath of Disease by Tamar Novik. It's in the Bulletin of the History of Science. And we have a link to that. Let's see what this link looks like. Redirecting. All right, we'll wait for the redirecting. With continuing thanks for all that my husband and I learned from This Week in Virology and other Microbe TV podcasts. Happy 2023. You have to buy it. It looks like.
No,
there's
a scan of
the
first page of it.
It
looks like
they want $25. Okay. Then P.S. Nothing to do with virology, but you might find it interesting anyway. By Karen reads, Vitamin B Complexities. Part of the series, What Should I Eat? Why? And gives a link to that as well. And Kevin writes, Hi, TWiV team. Greetings from upstate. I have consistently listened to TWiV and other Twix for over 4 years now. Your pedantic banter, quirky enthusiasm, article deep dives, and snippets and other intellectual interests and passions have influenced my life in very profound ways. You know, that sounds interesting, doesn't it? We should do a podcast with all that.
Yeah, we should include those things.
You really describe it nicely. Your shows have kept me company on road trips, entertained me during my morning commutes during undergrad, helped me find clarity in a mess of contradictory media, taught me basic virology and immunology, and then some in the absence of any courses, allowed me to find peace from learning about the molecular logic that allows microbial beings to dominate our macroscopic lives, and enabled me to have intelligent, founded, and empathetic conversations with many friends, family, and strangers about COVID vaccinations, treatment, and origin, all of which, statistically speaking, could have saved a handful of lives unbeknownst to me. Wow, that could be like the cover text for a book.
Yeah.
So sincerely, thank you, my friends. In the spirit of Giving Tuesday, I've sent a little bit of cash to Parasites Without Borders to be donated to microbe.tv. Thank you very much.
Thank you very much.
Kevin.
Thank you.
I'm now chest deep in my first year as a PhD student, forcing me into a new chapter as a Twix listener. Instead of listening to everything, I must pick and choose, which is a difficult reality to swallow. I'd like to suggest 2 YouTube channels as listener picks. The first is James Hoffman, a world-class coffee guru with frequent reviews and tutorials. Oh, that sounds good. And the second is Bald and Bankrupt, an unpolished world traveler and Soviet file. Their abilities to communicate clearly and infectious passion for their hobbies, all told in their splendid British accents, have made their channels number 2 and 3 in my favorites, just behind TWiV. Whew, good thing, 2 and 3. You can't compete with British accents, can you?
No.
I've linked my 2 favorite videos. I think you will enjoy them both. Thanks again, and I hope to see you live soon. No, live soon. We are going to have a live TWiV for number 1,000. I've decided with a few others that since we're all going to get together, this studio is too small. So I have found a venue in New York which seats 50 people.
Wow, you found a venue that big in New York.
And I'm going to try and book it. And so if there's interest, I'm sure there'll be interest. I've got
TWiV
30
at
people.
Madison Square Garden.
No, it's not going to be Madison Square Garden. But, I mean, there are some venues at Columbia I could look into, but we have some ideas.
Too much.
Columbia should give it to me for free, right?
Yeah, but it's Columbia, so.
Yeah. So anyway, this 50-seat thing could be cool. And I have some ideas for fun things which you're not going to learn about till you show up or listen to it. So live could be that.
Cool.
All right, that is TWiV 973. Is this the first one of that we've recorded this year? Yeah, it is.
Yeah.
2023. You can find the show notes at microbe.tv/TWiV. You can send your questions, comments, picks of the week to twiv@microbe.tv. And, we exist on your donations. So Microbe TV is a 501c3 nonprofit corporation which exists on your charity. So please donate so we can support everything. Microbe.tv/contribute. Alan Dove is at alandove.com. Turbotplaque.com he prefers. Thank you, Alan.
Yeah, either one. alandove.com is kind of my front page that people go to. You can find both sites from there.
Oh, they're 2 separate sites, right?
Yeah.
It's not just a redirect.
No. AlanDove.com, you'll get like a brief, like, cover letter kind of resume site, and then there are links to the personal blog and the science blog, and the science blog will take you to Turbot Plaque.
Got it. Brianne Barker's at Drew University.
And it's always a pleasure, by the way.
Oh, sorry. Yes. Thank you very much. Brianne Barker's at Drew University. Bio Prof Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here. I learned a lot.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWiV, and of course, all of you who support us. We thank you as well. I'd like to thank Jolene for the timestamps and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.